Hello students, welcome back. Welcome back to my biology online class. In my previous lecture, I had discussed with you about the detail of spermatogenesis, and from that knowledge we have already known that spermatid changes into a mature sperm by the process of metamorphosis. Today I am going to discuss with you about structure of human sperm. Structure of a mature human sperm. Sperm is haploid in nature. Haploid in nature means here chromosome number is N. This one is flagellate and this one is motile. So these are the basic information about sperm. There are 4 parts in human sperm. First one is head, second one is neck, third one is middle piece, and fourth one is a long tail. Okay. There are 4 parts: head, neck, middle piece and tail. Now I am going to draw the structure of a mammalian sperm, a human sperm. Let's draw the structure. First of all, the head portion, and the head portion has its 2 parts. The anterior one is known as acrosome, and the second part, second half of head is known as its nucleus part. Okay. Here will be gap. This one is acrosome and this one is nucleus, and both this acrosome and nucleus constitute the head portion of a sperm. Both this portion is covered by plasma membrane. This one is head portion. This one is head portion. After that, there will be a slight bending, and this one is known as neck, a slight depression. This one is the small one. This one is a short portion, is known as neck portion. After that, here this is a centriole, and here in neck portion there is also a centriole. This one is the neck portion, and these 2 centrioles, here one centriole is present, and in neck portion another centriole is present. This centriole, which is very close proximity to nucleus, is known as proximal centriole. This centriole is known as proximal centriole, and this centriole is known as distal centriole. Let's enclose them in a box. Okay. Axial filaments will be generated. Middle piece region. Middle piece region. This one is middle piece region. tail region is started. Tail region is started. This one is middle piece and this one is tail region. Here in middle piece, structure of spiral mitochondria. Let's draw this turning. Here to give it a difference with this axial filament, I have decided to use a separate color. This one is known as spiral mitochondria. And at the end of this spiral mitochondria, at the end of this middle piece, there are another two centriole. And this one is known ring centriole. These ring centrioles are two in number. Ring centriole. And this one is mitochondria, spiral mitochondria. And here 10 to 14 turn of mitochondria is present in middle piece, and at the end of middle piece, in both sides of this spiral mitochondria, two ring centrioles are present. Now this one is tail region, and this tail region has two parts. The first part up to here, this is known as main piece. Main piece. And this one is known as end piece. End piece. Okay, this one is PM, plasma membrane. This outer layer is known as plasma membrane. Beside this plasma membrane, there is also a membrane in inner side of this plasma membrane. But this one is not continuous as plasma membrane, as the plasma membrane is extended from head portion to the extreme tail end portion. This portion has a gap. Let's draw. Here it will going to cover the full acrosome part and nucleus part. Not all nucleus part. some of the anterior nucleus part. And this layer is known as Gallia capitis. Gallia capitis. Gallia capitis. Okay, and there will be gap, not continued. And this will cover almost 2/3 portion of head. After that, separate. Up to this portion. Here also let make this some thick portion. Continue this. Make it a thick layer. Continue and fill up this gap. And this one is known as manchete. Manchete, which will continue from this head portion, resting 1/3 of this head portion to up to main piece of tail. Okay. Now this diagram of the structure of a human sperm is very much important, and in your exam this diagram, if you have a question about draw and describe the structure of a human sperm, you should draw and properly point out this diagram and mention the title here, human sperm. Mention. Now all of we know the first part of the head is known as acrosome, and this acrosome layer here, the acrosome portion is covered by a sheath. First of all, from which the acrosome will be generated at the time of metamorphosis, when the spermatid metamorphosed into sperm. Process is known as all of spermiogenesis, spermatid metamorphosis. Okay, acrosome is made up of the Golgi bodies. All of you know, in our cell, Golgi body is an important cell organism, and the acrosome portion of the head of sperm is made up from originated from Golgi bodies. Okay. originated from Golgi bodies. This one is very important characters. Golgi bodies. The next point about acrosome, cap-like. Cap-like. And the cap, it is two-layered. Okay, the covering of acrosome is two-layered sheath. There is a two-layered sheath, and the frontal portion of this acrosome is known as cap, and the whole structure is cap-like. Okay, next is anterior portion of head. This is anterior portion of head. Head of which? Head of human sperm. So mind these 3 points very carefully. Okay. After that, there is a nucleus portion. Nucleus is posterior portion of head. Posterior portion of head. This one is spindle shaped. Spindle shaped, and the nucleus contains DNA plus proteins. Got it? DNA plus protein. Proteins means basic proteins. Basic proteins. So these are the two portions of human sperm head. One is acrosome, which is very important, structure. Nucleus is also important structure. It maintains. I am going to describe the structure first. After that, I will discuss about some function. Portion is neck portion. Neck portion is very short. Slight depression here at the depression of this nucleus portion, head portion, there is proximal centriole. If the question arises how many centrioles are present, there is basically two centrioles, proximal centriole and distal centriole, and there is two ring centrioles. Okay, so proximal centriole, where the proximal centriole is present? Proximal centriole is present towards the nucleus. Towards the nucleus. Distal centriole is present toward the middle piece. Toward the middle piece. And from this distal centriole, many axial filament is generated from this distal centriole. All of these known as axial filament. Axial filament of 9+2 microtubule arrangement. Axial filament of 9+2 microtubule arrangement give rise to axial filament. Give rise to axial filament. Axial filament will continue from this middle piece region to the main piece region of tail of human sperm. Okay, ring centriole is present where end part of middle piece. Okay, end part of middle piece. Ring centrioles are present at end part of middle piece. Ring centriole is also known as annulus ring, or Jansen's ring. Now come to this spiral mitochondria. Spiral mitochondria I have already discussed. There are 10 to 14 turn of spiral mitochondria, and this spiral mitochondria is also known as kern. Neighbor kern. Mind this spelling, Neberkern. Neighbor kern. Spiral mitochondria is also called neighbor kern. So this is the diagram and description of structure of human sperm. Discuss some function of this structure of human sperm. First of all, concentrate. Acrosome. It helps at the time of fertilization. Helps in sperm entry. And it releases enzyme. It releases an enzyme called sperm lysine. Mammal, it is known as hyaluronidase enzyme. Hyaluronidase enzyme. This enzyme helps at the time of fertilization. All of you know that the ovum has several layers. When sperm going to enter into the ovum, the first penetration is done by the hyaluronidase enzyme released from this acrosome portion of sperm. Come to the next portion. I have already discussed rich in protein, but mainly here as protamine. Sperm nucleus is haploid. Sperm nucleus is haploid. Now this proximal centriole is donated. It is donated to the egg at the time of fertilization when zygote is formed. All of you know at the sperm and egg, when sperm and egg is united, first of all a zygote is formed, and the function of this proximal centriole is to contribute and helps in forming the first mitotic spindle after fertilization. First mitotic spindle after fertilization. Okay, it gives, what is the function of this spiral mitochondria? It gives energy. It gives energy for motility of sperm, for swimming of sperm, and the sperm motility 1 to 4 millimeter per minute. 1 to 4 millimeter per minute. Now let's discuss a distinct point, the gap. Here we can see a gap between acrosome and nucleus of sperm head. This is a gap, and this portion is known as perforatorium. Perforatorium. The space between acrosome and nucleus of sperm head is known as perforatorium. This is a narrow fluid filled space. Narrow fluid filled space. Not all sperm has this tail structure. Some animals have no tail in their sperm. No tail in sperm. Ascaris. It has amoeboid sperm. Amoeboid shaped sperm. Crayfish. Star shaped sperm. Crab and lobster, spine shaped. And planaria, this has biflagellated sperm. So, some example which has no tail in their sperm structure. The speed of sperm after it is ejaculated in the vaginal tract. Speed is 1.5 millimeter minute. And after ejaculation to reach fallopian tube, the sperm takes 30 minutes time to reach fallopian tube. From vaginal tract, the sperm needs 30 minutes. Okay, human male ejaculate 200 to 300 million sperms during a single coitus, and of which for normal fertility 60% of sperms, 60% of all means 60% of these 200 to 300 million sperm, 60% should be in normal shape and 40% should be vigorous motility. 60% for a normal fertility, 60% should have normal shape and size, and 40% should have vigorous motility. Let's discuss some pathogenic condition. Aspermia. Aspermia means semen is absent. Semen is absent as well as sperm also is absent. And the next clinical condition, azoospermia. No sperm in semen. And what about necrospermia? Dead sperm in semen. These are all pathological conditions. So this is all in a nutshell the structure and function of a human sperm or a typical mammalian sperm. So stay connected. In the next class I will discuss about oogenesis.